Hello everyone, welcome to Climate Classes, and today's our topic is biotechnology and its application. So some of the curious things about this chapter is that biotechnology is a sector which deals with the production of pharmaceuticals and other biology. So using genetically modified microbes, and it also has immense application in various fields such as agriculture and also health. And this chapter deals with how biotechnology has contributed in development of medicine as well as agriculture. And coming to the weightage of this chapter, in meat, it holds around 4% of the weightage, and in boats, it carries 12 marks. Some of the topics to be covered are biotechnology application in agriculture, such as BT cotton, and biotechnology application in healthcare, such as genetically engineered insulin, gene therapy, molecular diagnosis, and transgenic animals and their uses, ethical issues. How has biotechnology contributed in the development of agriculture? And add a note on BT toxin and its contribution. First of all, biotechnology did contribute a lot in the development of agriculture. We're going to see that application of biotechnology in agriculture involves several techniques such as genetically modified organisms, BT cotton, pest resistant plants. All these sort of things are done successfully because of the application of biotechnology. And these are really important because in order to increase the production of food, these type of improvement need to be done for the crop so that the produce can be increased and the quality is also improved. And it helps in modifying plants, animals, and improve the agriculture productivity. Okay, several techniques like tissue culture, genetic engineering are also used. And coming to the BT toxin, BT meant for Bacillus thuringiensis. This is a scientific name. So it's a gram-positive spore forming bacteria. What exactly it does is that it produces, this Bacillus thuringiensis contains some cry and CYTG proteins. So what happens is that these releases some toxins which are consumed by the pest, and after entering the pest body, these releases the toxins. Okay, so these toxins are released by these bacteria will kill the pest, so that this way BT toxin is being helpful for the killing of the pest. So it is also used as biocontrol agent. So it is used as bioinsecticide to against the caterpillars, beetles, flies, etc. So, and write a note on structure of insulin. How is mature insulin different from pro-insulin? So, first of all, insulin consists of two short polypeptide chains. One is chain A and another one is chain B. These two are linked by disulfide bridges. What happens in mammals, including humans, is that insulin is not synthesized as a hormone. Instead, it is synthesized as prohormone. What it meant is that it needs to be processed in order to be a final and mature functional hormone. Okay, so insulin is synthesized as prohormone. It needs to be processed for becoming a fully mature and functional hormone. Okay, so here, and this contains an extra stretch called the C peptide. Okay, this C peptide is not present in the mature insulin. So the difference between the prohormone and the mature insulin is the presence of this C peptide. Okay, and is removed during maturation in insulin. So during the process of maturation, this C peptide is removed from the prohormone and is converted into the functional hormone. That's the difference between the mature hormone and the prohormone. So how is insulin prepared using recombinant DNA technology? What are its advantages? So for the synthesis of insulin, what happens is human insulin is excreted from pancreas cells, and an insulin producing gene is isolated. Okay, human insulin is first extracted from the pancreatic cells, and from those, insulin producing gene is isolated. Then from a bacterium, a plasmid DNA is extracted. A plasmid DNA is extracted from the bacterium and it is cut with the restriction enzyme. Okay, then forming this will act as a plasmid vector. Okay, this plasmid DNA which we have extracted from the bacterium is going to act as a plasmid vector. Okay, human insulin producing gene is inserted into this vector. Here we have extracted the human insulin producing gene, right? So we'll just insert this into the bacterial plasmid vector, okay, to form the recombinant DNA of the human insulin producing gene. So this recombinant DNA is introduced into the bacterial cell to form the recombinant bacteria. Okay, here first we are isolating the insulin producing gene from human insulin, and then we are extracting the plasmid DNA from the bacterium. This insulin producing gene is being inserted into this plasmid DNA and it is converted into recombinant DNA. And this recombinant DNA is introduced into the bacterial cell to form the recombinant bacteria. The recombinant bacteria multiply in a fermentation tank and produce the human insulin. Okay. Then this insulin is extracted, purified, and bottled. It is then ready to be injected into the diabetic patients. This is how the insulin is being prepared. Coming to the advantages, as a it does not require any sacrifice of animals such as pigs and cows, because there is no requirement of sacrificing these animals in order to produce insulin, because using biotechnological operations it's a quite a easy process to multiply the human insulin. It is safe and has least side effects. Okay, and the recombinant insulin can be synthesized in large quantities in a short period of time, and it is cheaper than that isolated from pigs and cows. It does not cause any immune rejection like animal insulin. And recombinant insulin can be used for insulin therapy for diabetes mellitus patients. It does not cause any allergic responses. So what is gene therapy? Explain by using an example. Okay. Gene therapy. Gene therapy is a technique which involves replacement of genes which are defective with the healthy ones in order to treat the genetic disorder. Okay, what exactly gene therapy means is that we need to identify the defective genes and we need to replace the defective genes with the healthy ones so that we will be able to cure the particular genetic disorders. So it's an artificial method that introduces DNA into the cells of human body. The first gene therapy was successful in the year 1989. So there are two different types of gene therapies depending on which type of cells are being treated. One is somatic gene therapy and then other one is germline gene therapy. So coming to the somatic gene therapy, what happens is here transfer of a section of DNA to any cell of the body that doesn't produce sperm or eggs. Okay, in this what happens is we are going to transfer the section of DNA to any cell of the body. Exception is sperm or eggs. Effects of gene therapy will not be passed on to patients' children. So here we are not going to transfer the section of DNA into any part or any cell that is going to produce sperm or eggs. So that this whatever we are just introducing into the human body, that is not going to pass on through generations. So germline gene therapy means transfer of section of DNA to cells that produce eggs also. Effects of gene therapy will be passed on to the patient's children subsequently. Whereas coming to the germline DNA, it is gonna be, I mean, like the DNA transfer is gonna, we are gonna transfer the section of DNA to those cells that are going to produce eggs and sperm, so that the effect of gene therapy is going on pass on through generations. How has biotechnology led to improvement in diagnostic modalities in medicine? As we know, as I said about human insulin, as normally it is it is normally if it is done manually without the biotechnology applications, then we are gonna use pigs, cows, and we need to sacrifice them and we need to extract the insulin from them so that it can be used for the diabetic patients. But what happens in this case is that because of using biotechnological applications, it made very easy for us, because there might be a chance of rejection because of animal insulin being injected into human body. it's quite a difficult possibility of rejection is more. So those can be avoided, and this can produce in a huge amount that in a very short period of time, which is really very good production of process, and no side effects will be there because it is just a multiplication of process. And coming to gene therapy, I mean, just the changes in the gene and replacing the defective ones with the healthy ones will correct the genetic disorder, and this can be passed on through generations. And molecular diagnosis, polymerase chain reaction. This is also an important application where biotechnology is being used, and pharmacogenomics. This is this in this includes the production of drugs, whereas edible vaccines, where we are going to take vaccines which are involves inactivated pathogens, right? So in all these things, the biotechnology is being widely used, and this really is improving the medical field. And I think biotechnology do have a huge application in the medical field apart from agriculture field. And now, what are transgenic animals, state their uses, and I don't know from transgenic crop. Transgenic animals are the animals with a modified genome. It means that a foreign gene is inserted into the genome of the animal to alter its DNA. In transgenic animals, what happens is that a foreign gene is inserted into the genome of the animal so that its DNA is altered. So this method is done to improve the genetic traits of the target animal. So suppose if I wanted to change some particular characters of suppose I'll take mice as an example. I just want it to be bigger, so that I wanted to insert a character or particular trait that I wanted to be in the target animal, so that I'll just take that particular character, which is a gene, which is rich in which do have that character, and I'm going to insert that into this animal so that it is going to alter its DNA and to produce the required traits. Okay, so transgenic animals are routinely used in the laboratory as biomedical research. So coming to transgenic cow, transgenic animals that produce useful biological products can be treated by the introduction of portion of DNA. So the first transgenic cow is Rossi, which was invented in 1997. It is helpful in the production of human protein enriched milk. The protein in milk was about 2.4 grams per liter. The milk has alpha lactalbumin, and it is a balanced food for human beings. So this is a transgenic cow which was produced, and which was helpful in the production of human protein enriched milk. This cow is do produce human protein enriched milk, which is about protein content in this milk is 2.4 grams per liter. It do contain alpha lactalbumin, which is food for the human babies. Coming to biopiracy. So, what exactly meant by biopiracy is that patenting and exploitation of bioresources of other nations without a proper authorization or access and benefit sharing agreement is called as biopiracy. Okay, biopiracy means that if suppose if we are using some other nation's bioresources without the acknowledgement of that particular nation is called as biopiracy. So it is being carried out by many organizations and multinational companies proposing that components of bioresources are products of their research. So some of the biopiracy related products are turmeric, neem, basmati rice, are some of the products have patents in other countries. So we do need to take, they are not our products, they have patents in other countries. Okay, write a note on golden rice. Okay, golden rice is a genetically modified biofortified crop. So what mean by biofortification is that improving the nutritional quality or value of the rice or any crop. So golden rice is one of the that kind that is genetically modified biofortified crop. So golden rice is genetically modified to produce beta carotene. Okay, it is modified to produce beta carotene, which is not normally present in rice. Normal rice do not contain beta carotene. So it is modified to this rice is modified to produce beta carotene, which is and beta carotene is converted into vitamin A when metabolized by the human body. Okay, so beta carotene is again converted into vitamin A when metabolized by human body. So we need vitamin A for healthier skin, immune system, and vision. So that's the purpose of producing or modifying golden rice. Okay, golden rice is genetically modified to produce beta carotene, which in turn produces vitamin when metabolized by human body. As we know, vitamin A is really helpful for humans for healthier skin, immune system, and vision. In its summary, as we know that biotechnology deals with production of pharmaceuticals and other biologics, and also immense applications in various fields. And I mean, its usage in the medical field, such as gene therapy, molecular diagnostic techniques like PCR, ELISA, regarding the transgenic animals and biopiracy and all these things. And about the institute regarding the admission and any other information details, you can contact the given number.